Our third speaker is Martin Hirst, who is going to talk about another program that is in partnership with CIHR, and this one is the Canadian Epigenetics Environmental and Health Research Consortium. So I presume that's linked to the International Epigenome Consortium. So great, thank you.
Great, well thank you for that introduction, and it's a pretty tough act to follow here, but I will do my best. I'm going to start my talk off today with really giving you the punchline, and that is what are the opportunities that present ourselves through the study of the epigenome. So at least from my perspective, the epigenome provides opportunities in personalized medicine. We just heard a beautiful example from Dr. Friedman about looking for genetic lesions in genes, but of course genes only make up less than 3% of the genome. And we know that genetic lesions occur outside, and those that occur within regulatory regions are likely to be disease causative, and the epigenome will allow us to look in those regions. We can continue that in terms of biomarker discovery and validation, and then utilize that in terms of genetic variant prioritization. It'll also allow us to provide insight in terms of basic mechanisms in genome regulation in normal, transitioning disease types, as well as provide novel therapeutic target discovery and validation. And there's really an emerging field of therapeutics directed towards epigenetic targets, primarily because they are potentially reversible, unlike genetic lesions, which are typically irreversible. So with that, now I'm going to give you a very brief overview of what is epigenetics, because I get this question all the time. So I'll start from the beginning. Of course, the term epigenetics, or the epigenome, came from Conrad Waddington actually in the 40s, before we even knew that DNA was the genetic material. And he was using it to describe the formation of the flywing, or what the process that gives genotype into phenotype. And so there are many facets to that, one of which we now know is the epigenome. And the way we like to look at it is that from a totipotent zygote, as we then differentiate down into the terminally differentiated cell types, we get a more and more defined epigenome. So what's come out more recently is that the epigenome appears to be plastic in some cases, and in fact responds dynamically to signals from the environment. And so I have this as this little lightning bolt. The environment then hits the black box, and this has effects downstream in, for example, the breast that I've given this green little star. Turns out to be a little bit more complex than that. We know that there are windows of susceptibility in organisms. This has been shown in epidemiological studies from humans, and I'm showing you this in the context of this northern Swedish town which studied feast and famine, and the amount of food that was available for the grandfather of a particular lineage would affect the lifespan of their grandson or granddaughter. But it was only during a certain period of growth, which is known as the slow growth period, that this feast of famine had an effect. So this is known as the windows of opportunity. And really, I suppose the surprising part is that we now know that this, at least in model systems, and this seems to be true also through epidemiological studies in humans, that this then can be passed on to further generations. So this insult happens to your grandfather, but this then gets passed down from the F1 to the F2 generation. So there's been a change in the epigenome that is hereditary and passed down through the generations. So what is the epigenome? Well, the epigenome consists of modifications to the chromatin and to DNA. So that's DNA that's packaged within the proteins that do the packaging. DNA, as we like to think of it, or genomes, are not linear. They're packaged very compactly, and this packaging involves a series of modifications, over 100 of them, to proteins and also to DNA in the context of DNA methylation, to proteins, DNA hydroxymethylation, as well as modifications to the histone tails. So these are the proteins that do the packaging. So those are the main types of epigenetic modifications that we study. We can also look at the three-dimensional structure of the genome through DNase 1 hypersensitivity. And more recently, we've now become to understand that actually non-coding RNA is playing a very dramatic role in regulating the epigenome by targeting certain epigenetic modifiers, such as the PRC1 complex that contains EZH2, or PRC2 complex, pardon me. So just to get a little bit more detail, so there's about 28 million CPGs, so this is a cytosine followed by a guanine in your genome. The majority of those are methylated, so they contain a methyl group, carbon and hydrogens. Gene bodies are largely methylated, but the methyl groups across the genome are not evenly spaced. They come in clusters known as CPG islands, and these CPG islands tend to be in regulatory regions up front of genes, and when these are methylated, the gene is turned off. So you can turn a gene off with a genetic lesion leading to the loss of function, or you can turn a gene off epigenetically. And we now know that, at least in the context of malignancy, that the genetic and the epigenetic regulations collude together to bring about disease. So in specific regulatory context, methyl CPG is repressive, as shown there. So in terms of histone modifications, we can broadly group those into those that reinforce active chromatin states and those that reinforce repressed chromatin states. Chromatin being the histones and the nucleosomes that pack the genome. And the ones I'm listing here are the ones that are part of the roadmap core and the ones that are part of the International Human Epigenome Consortium, which I will introduce in the next few slides. And these are the ones that are commonly studied. So marks such as H3K4 trimethylation, and this is the nomenclature of the epigenome, where the first indicates the histone, the H3 histone. The second indicates the amino acid, lysine 4, so this is the fourth lysine on the N-terminal tail of the H3. And then the last indicates the type of modification, so 3 methyl groups, so H3K4 trimethylation. Methylation can come in mono, di, or triforms. We also study the monoform that marks enhancers, etc., or as indicated. We also study repressive marks, such as H3K9 trimethylation, which is associated with repressive or heterochromatic DNA, as well as the polycomb group H3K27 trimethylation. This is laid down by EZH2. So what are some epigenetic effects? I've mentioned one. What are some other that we know that suggest to us that the epigenome is playing a role in disease? Well, studies such as the Dutch hunger winter, where caloric restriction was introduced through the Second World War. Women that were pregnant during that period of time gave birth to babies with very low birth weight, not surprisingly. As those babies grew up into adulthood, they had a much higher incidence of cardiovascular diseases, psychiatric disorders, etc. And the children of those children, so the grandmothers who actually experienced this insult, are also showing this sign of increased disease preponderance. If we look at fraternal versus maternal twins, we can see that some traits seem to be very tightly genetically linked. For example, height. So here's showing height, in the white bars being the maternal, the gray bars being paternal. So you can see that almost all of the disease burden can be characterized by the genetic. But as we go down into some more common diseases, such as cancer, breast cancer, diabetes, Crohn's disease, stroke, the genetic linkages are very, very weak, suggesting that there's more of an environmental influence. And of course, some of us believe that environmental influence is being patterned through the epigenome. And more recently, with work from Jamie Kramer and others, have been showing that in fact the epigenome seems to be playing an important role in mediating memory. So when we knock out, in this case, G9A, which is H3K9 dimethylation, and this is in a fly model, you can actually eliminate long-term memory, or LTM, and this is in a courtship model in this particular case. So it seems to be affecting a wide variety of both diseases as well as brain functions. So another important discovery that has come out in the last 3 or 4 years, primarily on the back of the explosion in our capacity for sequencing genomes, is the realization that there are mutations to epigenetic modifiers or recurrent observations in cancers, and in particular in hematopoietic malignancies. So Dr. Friedman spoke of EZH2, which is a discovery that came through the Genome Sciences Center in tight collaboration with clinicians, Randy and Joe and others, where we showed in 20% of diffuse large B-cell lymphomas there was an activating mutation in EZH2. But this has been shown in a wide variety of other hematopoietic malignancies, including leukemias, And of course, there are now quite a number of efforts out there to develop drugs against these epigenetic modifiers as a way of being therapeutic targets. So, for example, just recently in Nature, GlaxoSmithKline produced an inhibitor to EZH2 to be used in the context of lymphoma. There's an inhibitor to specific acute myeloid leukemia gene fusions, actually not to the gene fusion itself, but to the enzyme called DOT1 that's recruited and actually does the methylation of lysine 79. And I've color-coded these as being either active or repressed. Acute myeloid leukemia is kind of an interesting case where we see mutations to a variety of epigenetic modifiers, but they all appear to be playing a role in DNA methylation homeostasis. And so this is data that's come through Ali Carson's group in the Acute Myeloid Leukemia Personalized Medicine Project that's funded through Genome British Columbia. And I've indicated 4 genes here, IDH1, IDH2, TET2, and DMT3A, all of which are involved in mediating DNA methylation homeostasis. I don't have time to go into the details, but just to show you that, hematopoietic malignancies involve mutations to epigenetic modifiers. So what is the technology shift that's enabled us to start to look at the epigenome? Well, really next generation sequencing. And in fact, the first libraries that were sequenced on NGS platforms, such as Solexa at the time, or Solid, or even to some degree 454, were chip sequencing libraries or epigenetic libraries. And so just to sort of go through, really the seminal paper of Feinberg and Vogelstein back in 1983, showing this characteristic hypomethylated state of cancer genomes. All cancer genomes are characterized by a large loss of methylation, DNA methylation. This was using a restriction enzyme mapping, so they digest either the normal, as indicated in S, the N, or the carcinoma. This is an adenocarcinoma in C. And then they would use a radioactive probe, and by seeing the shift down, that indicates a hypomethylated state. With the advent of next generation sequencing, we can now do this at a single nucleotide level. The top track here in this genome browser shot shows methylation. Each of those bars indicates the methylation status of a single CPG in the human genome. We can do this for all 28 million genomes. This was actually the first whole genome methylome that was done, the top track. The bottom being a differentiated cell type. And now through the efforts of consortiums such as the International Human Epigenome Consortium, we can now do this for a wide variety of different cell types. And we can indicate in this super track on the bottom regions of the epigenome that are constant across many cell types, in this case 12, and regions that are variable, that is highlighted in the blue. And those variable regions are the ones, of course, that we're most interested in studying. Okay, so with this invention of the technology and our applications of it has come the ability to do large-scale epigenome mapping. And what came out of that was the birth of the International Human Epigenome Consortium. And there are efforts around the world who are contributing to this effort. The founding member was the NIH Common Fund, Reference Epigenome Mapping, which I'll be talking about. The Canadian initiative, the CERT platform. Blueprint, which is the European Union's version. There's a Japanese, a German version called DEEP. There's one in South Korea, and there are others on their way to being funded. And there's the link if you'd like to see the website. So what is the challenge in terms of generating reference epigenomes? Well, unlike our genomes, where we basically have one per organism, although there are some cell types that are variant, we have many epigenomes. So every cell type potentially has a different epigenome. So what we are attempting to do is generate reference epigenome maps for the majority of human cell types to act as a foundation to drive research and disease understanding forward. And there are some who are trying to understand whether, for example, PBMCs, or peripheral blood, which is a tissue type that is readily available, at least clinically, can be used as a surrogate in some cases. So people such as Mike Kilgore working in CMT, for example. So what we're trying to do really is generate reference maps that can be used as counterparts to complex diseases, such as reference maps for the immune system, to look at asthma, airway cells, autoimmune, etc., etc. So that's really the overall goal. And the overall goal of the IHEC is to generate 1,000 reference epigenome maps in the next 5 years. So the NIH reference epigenome mapping has been going on for 4 years. This project comes to an end June of this year. We've been involved in this project through our collaboration with Joe Costello at UCSF. These are some of the cell types, at least in the adult, that have been profiled to date. We're somewhere in about 150 reference epigenomes have been generated to date. If you're interested in accessing the data, it's listed here. And there is through Washington University, and Ting Wang has generated a very nice epigenome browser for browsing this data. The Blueprint Consortium, led by Henk Stenenberg in the Netherlands, is focusing on blood cell types primarily, and aims to generate reference epigenomes from 50 normal and 50 neoplastic blood cell types over the next 5 years. This is funded through a €30 million initiative through the EU. And then the Canadian initiative, which I'm here to talk about, is the Canadian Epigenetics Environment and Health Research Consortium. This was really started in 2011 with the RFA for the Epigenomics platform, as well as a Catalyst grant. This was followed up by an Epigenetics fellowship, and then the team grants, which are still under review, as well as another team grant involving Canadian and Japanese collaborations. So the platform that I'm going to talk about today was funded in April of last year, in 2012, so we're less than a year into the project. So what are the two centers that were funded? There's one in Vancouver and there's one in Montreal and McGill. The one in Vancouver is led by Dr. Mara and myself and Dr. Jones co-lead. It's funded through CIHR and Genome British Columbia. Our targets are primarily in the context of malignancies. So we are trying to match normal and malignant cell types and generating reference epigenomes for both. And so you can see the list of genes that we're looking at. The ones that I've starred are tissue types that are currently in the pipeline, including peripheral or lymphoid B cells, mammary epithelium, and colonic epithelium. The other center, which is led by Dr. Lanthrop, Meany, and Pastine, is funded through CIHR and Genome Canada. You can see their targets. In addition to the mapping platforms, there are also 2 epigenomic data coordination centers that were funded, and these will be involved in generating high-level analysis of the data and providing the data to the research community, both within Canada as well as within the world. So the aims of our center are to first establish the center and then to generate high-quality human cells for comprehensive epigenome analysis, and finally drive epigenome mapping technology development. And I don't have the time to really go through these in any big detail. We, of course, like Dr. Friedman, critically rely on our clinical colleagues, and so the tissue that we obtain comes through collaborations, many of which are long-standing with clinicians within the BC Cancer Agency and outside, and they're listed here. I'm not going to go through the details, but I'd be happy to talk about it with you after the talk. What is a reference epigenome? It's essentially chip sequencing for 6 core histone modifications that are listed here, as well as DNA methylation profiling by sodium bisulfite sequencing. We, within our center, are developing oxybisulfite sequencing to capture hydroxymethylation as well. And we're also doing whole genome shotgun sequencing, at least on a subset, to try and understand a little better the interaction between genetic and epigenetic patterning. We're also doing RNA profiling, both small RNA, long non-coding RNA, and message RNA. So I'm just going to go through, in the interest of time, a couple of vignettes. I don't know how much time I have left. I'm going to go through a couple of vignettes very quickly, just to give you a feel for what types of analysis are enabled with this kind of data. So what I'm going to talk about are looking at mammary cell types. In this particular case, these are breast cell types that were generated in collaboration with Thea Tilsey's group at UCSF from reduction mammoplasty. So these are phenotypically normal, disease-free human subjects who come in for a cosmetic surgery. The cells are then sorted for either myoepithelial, luminal epithelial. So myoepithelial are the cells on the outside of the duct. Luminal epithelial are the inside of the duct, and in appropriate hormone conditions produce milk. And then progenitor stem cells, which lie within the myoepithelial layer. We can sort those cells using cell surface markers to greater than 95% purity, and then do epigenomic profiling. Here I'm showing DNA methylation, so by simply looking at the DNA methylation state of, in this case, CPG islands, we're able to recapitulate the clustering of the cell types by their DNA methylation patterning. So stem, myelin, luminal. We can then look closer at regions that are defined by a high methylation content in either the stem on the right-hand side or the luminal on the left-hand side, methyl being red, non-methylating being CPG, so we can start to zoom in. And then we can actually look at specific genes. In this case, I'm showing you GATA3. We can see that GATA3, so the bars in this genome shot, the black bars indicate methylated CPGs, the gray bars equal unmethylated. So we can see that in this case, GATA3 is heavily methylated in the progenitor population and becomes epigenetically de-repressed in the myoepithelial and the luminal population. So this is the bipotent progenitor, gives rise to the myelin and luminal. So we can also try to do this on much larger data sets. And here's data generated through the reference epigenome using really an algorithm that came out of the ENCODE Consortium, for those of you who might know ChromeHMM from Manolis Kellis's lab and Jason Ernst. We can take the data, which is indicated here, so here are the marks, and we can ask, can we use a hidden Markov model to look for patterns of chromatin states that are defined by these particular emission parameters, emission being the presence or absence of that mark. So for example, at the very top, the transcriptional start site in the poised position is indicated by the presence of H3K4 trimethylation and H3K7 trimethylation. And then we can ask, well, how do these genomes look? And so we can do this across, in this case, 92 genomes. So I'm showing you here, and these colors indicate the different chromatin states. And now we're really looking at the functional genome from an epigenetic perspective. And what I want you to draw your attention to is, in the middle here, this is gene PAX5, a gene that's a marker of B cells, at least late B cells. And you can see, just by looking, this actually is the CD19 cell. So this indicates an actively transcribed promoter driving PAX3, or PAX5 in this particular case, a transcription factor driving B cell development. But we can also look a little closer, and so now I'm showing you some of the cell types. So you can see certain things. For example, on the right-hand side, here's a novel non-coding gene. We can see that it is patterned by the epigenome, so it's actively transcribed, giving us further support for the further evidence for this non-coding gene. And we can also see added granularity. So in these skin-derived cell types, these are blanketed by gray, which is actually a heterochromatic mark. So these are heterochromatically repressed in the skin cell types, but in other blood cell types, they're actually in a quiescent state. So they're neither active nor repressed, but these T cells seem to have this quiescent state. So these are the types of analysis that are mediated by this analysis. And just to sort of make the point, when we then go back and look at, I'm now looking at the example of the breast data again, we can use these chromatin state segmentations, overlap with the DNA methylation data, and we can find hypomethylated enhancer regions that are indicative of transcription factor binding, and we can then overlap those with whole genome shotgun variants that lie outside of coding regions to look for functional consequences or to prioritize these genetic lesions. So in summary, I just hopefully made the point that epigenome deregulation is implicated in human disease, including cancer. International investment in reference epigenome profiling is developing a framework for epigenomic research, and I've calculated it's on the order of about 400 million. And epigenome profiling in malignant cells is feasible, and we are beginning early efforts as well as many others. So finally, just to acknowledge the people that are involved, including at CHR, Eric Marcotte and Anthony Phillips, who've been invaluable at driving this project forward. So thank you very much.